would like to share with you. And finally, I will conclude the lecture. And those who have no idea about IHR is the biggest horticulture institute in Asia and mother of many horticulture institute carved from this institute. And more than 150 scientists working on different aspects of horticulture crops. And last year we had a very successful horticulture fair, national horticulture fair, wherein 55 lakh people participated offline and online, and 22 countries participants were benefited from this horticulture fair. And we also celebrated our golden jubilee in the year 2017. So it is an institute which has contributed a lot for the development of the horticulture in India. So coming to the diversity in horticulture, all of you agree with me, horticulture is a big area wherein we have fruits, vegetable, flowers, spices, medicinal aromatic plants, coconut, the plantation crops. So it is difficult to deal with this diversity, and difficult to conserve these genetic resources. And most of the crops in the international jargon, it is called orphan crops. Not much information is available, and the genetic resource collection, exploration, collection, and conservation is a difficult job. And you see here in this slide, the share of India in the world horticulture trade is given. We are contributing only to an extent of 2%, and the rest of the world is contributing the other 98%. And though the production of horticulture crops exceeded the production of cereal crops, but our contribution is abysmally low. And if you come to the global issues identified in horticulture, the genetic resource conservation and development is taken as one of the key issues in the issues at global level. And now what is happening is we have to feed the billion population, and there is a population explosion. And with the pandemic, now the food basket is changing, and horticultural crops are adding value for the food, and food has become medicine. And another issue with the production is the climate change. For an increase of 1 degree centigrade average temperature, there is a decline of 0.2% decline in the agriculture production. So this also adding a problem for the production of food for the people. Anil? Yeah. And the approach for the conservation is like this. Now, as I told you, food has become medicine. Earlier, we used to take food as a food and medicine as medicine. Now that paradigm is changed, and we are now prospecting for high yielding source of non-metabolites in different food species. And initially, we should locate the populations and develop the population atlas, and develop the chemical profile of the species and population, and developing genetic profile, then go for the conservation. So now most of the institutes are working on this line, that we see the profile, the chemical profile of the each food we are taking. Earlier we were not doing that. Now the total characterization is a must for getting the benefit of the food we are taking. And coming to the conservation strategy for any taxa, this slide I have taken from a classical paper written by Maxted in 97. So we should identify the taxa, then the conservation objectives to be laid down, then we should do the field exploration to collect the diversity. Then comes the conservation strategies. For any conservation strategy, the idea is to conserve maximum genetic diversity. So we should have different methods, because if you use only one method, there is a chance that we are not able to conserve the entire genetic diversity for a given species. And what I mean is the gene pool of that species. So we should do with seed storage, and if a seed is not available, we go for the field gene bank. Then for the other species, we go for the botanical garden, then in vitro storage, as Dr. Anita rightly mentioned, then pollen storage, DNA storage. Of course, the ex situ conservation, now the strategy of conservation or the concept of conservation is also getting changed. And ex situ conservation is no longer considered as the best for conserving the genetic diversity, and we are moving to the in situ conservation. But in India, in situ conservation for the cross species is not practiced in a big manner. And now the NBPG, some of the scientists and the director is trying to do the in situ conservation for at least for some of the cereal crops like rice. And for the horticultural crops, it is still difficult to conserve the diversity in situ form. So in situ conservation can be done by genetic reserves, on-farm conservation for the cross species, and home gardens. And once you have the conserved material, and finally we should able to utilize the material which are conserved. And in short, we can conserve the whole plant seed. If seed is available, then if it is vegetatively propagated, we will go for the in vitro conservation using the tissue culture techniques, and of course pollen and DNA also. Recently, even the conservation using DNA is also possible, and there are many methods available where we can use this DNA for can be inserted and the trait can be expressed. So in short, we have two types of conservation strategies available for us for conserving the genetic diversity for a given crop, and that is in situ and ex situ. And for the agriculture and horticulture crops, we go for the ex situ conservation, and in situ conservation is good for the wild species, wild relatives, land races, and traditional varieties. And normally the in situ conservation is practiced through the biosphere reserves, national parks, gene sanctuaries, and on-farm conservation for the agricultural crops. And recently NBPGR was having a MOU with Botanical Survey of India, and in that we are planning to have the collection of the wild relatives from the national parks and biosphere reserves. It is difficult to get permission to explore the national parks and biosphere reserves because it is with the forest department and with the Ministry of Environment, Forest and Climate Change, and they are restricting the entry. In fact, now for a National Medicinal Plant Board sponsored project, we are looking for the permission from the Forest Department. We are yet to get the permission from the Forest Department. And ex situ conservation is normally practiced by establishing the seed gene bank, and the seed gene bank is the best form of conservation. But the issue is most of the horticultural crops are having the recalcitrant seed, or seed is not produced, vegetatively propagated. So we should go for the in vitro conservation or cryopreservation, and cryopreservation is the only method available for long-term ex situ conservation. And of course DNA bank is also a form of conservation. So we can have the crop gene bank, arboretum, herbal garden, and botanical garden for conserving the other species. So the different types of conservation is seed conservation, in vitro, then field gene banks, and for the clonally propagated species and rarity species, we go for the field gene bank or in vitro gene bank. So in vitro, by using in vitro gene bank, you can have the medium term conservation using slow growth in vitro. and cryogene bank for long-term conservation using cryopreservation. But the issue with the cryogene bank is you have to develop a species-specific conservation cryopreservation protocol, and that is available only for 200 species. And for each species, we should develop a specific protocol. And for a tropical species, conserving material in using cryopreservation is a difficult task. Now, I will go to a little bit detail about the different conservation strategies. As I mentioned earlier, the for any conservation strategy, the aim is to conserve the gene pool or maximum genetic diversity of that given species. And conservation is a process that actively retains the diversity of the gene pool with a view to actual or potent utilization. And if you are not able to utilize, then there is no meaning for the conservation. And the utilization is the human exploitation of the genetic diversity. And the ex situ conservation, the definition is the conservation of components of biological diversity outside their natural habitat. And in situ conservation is exactly opposite. It means the conservation of the ecosystem and natural habitat. And ex situ conservation is minus evolution, and in situ conservation, the species is getting a chance to evolve. And that is very important in the given circumstances that we are facing the climate change. So if we are not conserving in situ conditions, the species may not be able to survive in the changing environment scenario. Ex situ conservation is a complementary approach. See, now, as I mentioned, with one conservation strategy or with one technique, we may not be able to conserve the entire genetic diversity for a given species. So we should go for the complementary conservation strategy, and that involves more than one technique. So now, again, these are the different techniques available for conservation, that is ex situ using seed storage, then in vitro DNA, pollen, field gene bank, and botanical garden, and in situ using genetic reserve, on-farm and home gardens. So in the ex situ conservation techniques, genetic variation is maintained away from its original location, as I mentioned. And these techniques are generally appropriate for the conservation of crops, which we are dealing, especially for horticulture crops. Again, problem there. And field gene bank is one form of conservation strategy wherein the horticultural genetic resources, especially the tree species like mango, can be preserved. And you can see here our field gene bank of mango conserved. And the advantage of field gene banks are it is suitable for species producing recalcitrant seeds, and it is available for easy characterization, evaluation, and utilization. But of course, there are disadvantages like the material is susceptible to pest diseases and vandalism, and involves large areas of land, and a lot of inputs are required, and high maintenance cost is a problem for maintaining the field gene bank. And there is a chance of getting the material is getting eroded in the gene bank, field gene bank also. Then in vitro storage, especially for the vegetatively propagated species, and the advantage is it is in a sterile and pathogen-free environment. And for banana, it is the ideal technique for conservation. And the Biodiversity International is, they are exchanging the material only in the form of in vitro cultures. And you can see on the right side, the International Potato Research Institute's in vitro gene bank. The advantage of having in vitro bank is relatively easy for long-term conservation for recalcitrant sterile clonal species, and easy access for evaluation and utilization. Of course, there are disadvantages like risk of somoclonal variation, need to develop individual maintenance protocol to be developed, and relatively high-level technology and maintenance costs. So these are the reason why many countries are not able to go for the in vitro gene banks. Then seed storage, it is the best form of conservation, but the issue is many of the horticultural crops are not producing orthodox seeds, and it is efficient and reproducible, feasible for medium and long-term secure storage, and wide diversity of each target taxon can be conserved, and easy access for characterization. And disadvantages are problem of storing seeds of recalcitrant species. See, recalcitrant species, the disadvantage is you cannot reduce the moisture content, and without reducing the moisture content, you cannot store it at low temperature, and it freezes the evolutionary development, as I mentioned earlier, and genetic diversity may be lost with each regeneration cycle. After 20 or 30 years, you have to regenerate it, otherwise the vigor will go down. So in each process of regeneration cycle, the genetic diversity of a given species is getting lost. So these are the issues with the seed conservation. And conventional seed storage is a safe, effective, and inexpensive method, and you can prolong the seed longevity for 50 to 100 years. And the optimum stage of seed maturity is lot quality, processing, and harvesting techniques. All these are important. And as I mentioned, orthodox seed can tolerate drying. Then recalcitrant seeds are damaged by desiccation. Intermediate seeds are seeds that do not fit. There are three categories of seeds: intermediate, recalcitrant, and orthodox. And recalcitrant and intermediate seed is difficult to store at a low temperature. Then coming to the pollen storage I will be dealing separately. So DNA storage, relatively easy, low cost conservation, and there are disadvantages. From DNA you cannot regenerate the plant, and from pollen also you cannot regenerate the plants. Now cost is a very important issue for conserving the genetic resources, especially a country like India. The resources are limited, and with the pandemic, we are facing problem of funding for many projects are not available. Many projects are not getting funded. Now, field in vitro and seed for wheat and maize, compared, and these are all from the CG centers, that the consultative group of the National Agricultural Research. And you see the total cost for accession for field gene bank, it is $17.909 US. For in vitro is maximum, that is more than $26. And for seed, for wheat it is very less, and for maize also it is less. So it is better to conserve seeds, but the problem is whether seed is available or not. Then you may ask the question, what are the benefits of conserving the genetic resources? This slide shows the benefit of conserving the genetic resources of different types of horticultural crops. You can see here, for the pharmaceutical industry, botanical medicine, major for crop protection, biotechnology, and cosmetics. And the lowest estimate is 500 billion US dollars, and the higher estimate is 800 billion dollars. So a huge benefit we are going to accrue if you conserve the genetic resources. And as I mentioned, for most of the horticultural species, we should go for the complementary type of conservation. One technique or one method is not going to serve our purpose, so we should go for more than one method or strategy for conserving the genetic resource to get maximum genetic diversity conserved in a given space and time. Now, in a complementary conservation strategy, see, complementary conservation strategy is using more than one method which is suitable for conserving one species. If you take the orthodox seed, root crop, and forest species, by using different methods, how you can conserve maximum genetic diversity, that is given in this slide. And you can see for an orthodox seed species, seed storage, with the seed storage you can conserve maximum genetic diversity. But in the case of root or tuber crops, you have to go for the field gene bank. And forest species, we should go for the forest reserve. And by using more than one technique, 5 or 6 technique, you can conserve the maximum genetic diversity available in that species. And we were having one project under UNEP Global Environment Facility, administered by the Biodiversity International. in that project, we evolved, strategy for conserving the various fruit species like mango, Garcinia, citrus. And in that, and I consider this project was very successful. Why? We could identify 83 custodian farmers, and 180 self-help group was formed. In fact, most of our conservation strategies, the all stakeholders are not involved. Unlike the other projects, this project involved all the stakeholders, and because of the involvement of all the stakeholders, this project was very successful, and we could identify the custodian farmers, and they were honored in different platforms, and they were very happy. And 27 fruit catalogs were developed, and 61 diversity fairs were organized, and a lot of products were made, and 140 training programs. And in addition to this, very good technical bulletins, research papers came out of this project. In fact, I consider this project as a model for the conservation of tree horticultural crops. And now I will just ask your attention for the world scenario of conservation of plant genetic resources. And when it comes to the world scenario, we have to see the CGIAR gene banks, that is consultative group on international agriculture research. They are having 11 centers together, maintain 7 lakh samples. And there are horticultural crops involved in that. I will give you the information in the coming slides. And you see the location of these gene banks. We are having this ICRISAT at Patanjuru, Hyderabad in India. Rest of them are in other places. And you see here the cassava, then bean, and tubers, potato. These are all the horticultural crops, and the number of accessions are given in this. Again, cassava, cowpea, but number of horticultural crops are very less with the CGIAR centers. So a total of 532,508 accessions stored in the gene bank of CGIAR institutes. Now, with this background, we will go to the our work, that is the conservation of horticulture genetic resources at ICRI-IHR. So the activities which I am going to show you are the exploration collection missions, only a glimpse I would like to show you, and establishing maintenance of pollen grave bank, which is our mainstay, that it was featured in the Limca Book of Records, and now we are having more than 700 accessions of pollen belonging to 100 species of 50 belonging to 50 families. Then I would like to show you a case study on developing in vitro system for the Northopoditis pneumoniana and field gene bank of RET medicine plants. And finally, I would like to show you a case study on Maduka insignis, which is very rare plant. And now coming to the conservation, the exploration collection of genetic resources, where we can collect this material which is having value, traditional material and land races. You can see here we were in the North Kannada region, and there the tribes were there, and you can see the Gauli tribes. And when this exploration was in collaboration with the NBPGR, Dr. Joseph John was also there with me. And these Gauli tribes were not at all, ready to share the material or information with us. But when we went with our vehicle, all the children were seeing what we were doing. And this is a very interesting tribe. This is called Siddhi tribe, and these people came along with the Portuguese 500 years back, and Portuguese left us. These tribes were living in the North Kannada region, and you can see their home, and they cultivate a lot of vegetables, and they were ready to share the material with us, and the information also they were sharing with us. And here very interesting information. You can see on the left side a brinjal variety which is weighing 500 to 750 grams, and it is getting cultivated in Shimoga, and you can see the farmer on the right side below. And you see the sod beans is cultivated by the Gaudi tribes. And for Amaranth, we could collect 45 accessions belonging to 7 species. Very good variability we could collect, and you can see there are grain amaranth, there leafy vegetable, and you see the colors. A lot of diversity we could collect from the. And this is from Maharashtra. There in Ratnagiri district, in one area they were cultivating this vegetable cowpea and brinjal only in a very small place, but it is traditional varieties. And Dharwad, we did an exploration there. We could collect the local land races of brinjal, like Bettigare local, Pudichi local, Lopur local, and Malapur local. For brinjal, we are having more than 3,000 varieties, and these are all having GI tags, and some of them already the Shimoga and they registered this as a GI. And we had been to Uti Uttarakhand for collecting the French bean diversity. This came along with Europeans, and we could collect 38 accession of French bean. Then in Goa region, we could collect the Momordica sahyadrica species described by Dr. Joseph John. You can see he is along with that plant. And we could collect the diversity in local Kukumis sativas also. Then in Dashin Kannada, we could meet one farmer called Monappa Karkari is having his own farm. And another strategy they were having, were having the community seed bank. If you go with your material there, you can exchange the material. So that is a good concept, and the farmers are using that concept to conserve and use the genetic diversity, especially in vegetable crops. Then coming to our field, our pollen cryo bank, it's the biggest pollen cryo bank in the world, and now we are having 100 species, as I mentioned, and we have successfully completed 37 years of cryo preservation, and the maximum period we tested was 26 years. And this involves technique for collection, viability, and fertility assessment for 100 species. And as I mentioned, it is featured in the Limca Book of Records in 2001. And this is our pollen cryo bank. You can see the, we are conserving this material in this cryo bank. And I would like to tell you this technique has been transferred to 5 seed companies all over India. The first one was VNR Seeds, then to Rasi Seeds, then Indo-American Hybrid Seeds. Then this year we transferred to 2 seed companies like Seedworks and the Nanams. So these people are using this for the hybrid seed production in vegetables, especially in the solanaceous vegetables. They are facing the problem for how to how to conserve the male parent, that is pollen. In fact, they were using these farmers for getting the hybrid seed for hybrid seed production, but the farmers are becoming very smart and they themselves start producing hybrid seeds. So now the companies are having this technique for getting their male gamete without poaching. They can conserve them. So this is our seed bank. You can see this. With 1 crore rupees, we refurbished the seed gene bank, and this is the in vitro bank. And now coming to the field gene bank for rare and endangered and threatened species. This we got a project in 2008 from National Medicinal Plant Board for conservation and conservation of 32 rare and endangered threatened medicinal plants, and IHR was the lead center, and other 9 institutes were also involved, and we were the lead center, and we established this field gene bank. And you can see the different view of the field gene bank which we established, and this is completely organically maintained, and We are not using any chemicals for maintaining this. Now we have completed some 11 years, and if anybody is interested, you can come and see. It is in a very good form, and there are so many rare plants we could conserve in the field, and they are flowering and fruiting, and we are getting the seeds from these plants. And those RET medicinal plant, we did the chemical profiling. We would like to know what it contains, and we went for the chemical profiling of this plant, and you can see the same chemical profiles of these medicinal plants here. Then we did. We would like to know what is the diversity available in these plants, and for that we did the molecular characterization, and you can see the we use the ISR markers, and you see here the Acorus calamus, Elastus paniculatus, Alpinia calcareta, and Oryxellum indica. And we got a string of visitors. Every year we are getting. You can see Dr. Pushpanathan. He was former director of JMT BGRI and NBRI, and Dr. Sanjappa was a former director of Botanical Survey of India. And now the next case study is on Nothopoditis pneumonia. It is also the synonym is Mapia foetida, and this plant is considered very important. It is the chemical is extracted that is used for the treatment of cancer. And we had a project from National Medicinal Plan for this plant, and it was very difficult to establish the tissue culture system for this medicinal plant. And except the excised embryo, no other explants were responding, and it took 6 months for one of my students to establish the in vitro system. But finally, we could establish the in vitro system. You can see in this slide, and then we could develop the somatic embryogenesis in this plant after 3 weeks of incubation, and globular structures were developed. And somatic embryos to the tune of 17 numbers. Then you can see emergence of the plants from this somatic embryos, and you can see the. Then the other way also we did, that is induction of multiple shoots, and without TD set we were not getting the response, and TD set induced the shoots, and even though TD said that thidiazuran was promoting somatic embryogenesis, adventitious shoot formation, shoot elongation was not satisfactory. And root you have to induce separately. For that we used 1 mg per liter of IBA. Then we went for the in vitro conservation using reduced culture medium, that's low light and low temperature, and we could conserve for 6 months these plantlets. And we went for hardening also, and you can see here we could establish the plant in the pot. Now, in the next 5 minutes, I would like to share our experience in conserving a very rare plant that is called Maduca insignis. And this project was sponsored by Department of Biotechnology. In fact, it was in a network mode. There were 38 organizations were involved in All India Network project sponsored by DBT, and it was very interesting. When we started this project, we were not knowing what is this plant. I have never seen this plant. Then we asked some few questions. What is the current status of Maduca insignis? So what we did is, first you can see in the IUCN Red List, International Union for the Conservation of Nature and Natural Resources, and it is mentioned that extinct, and it was rediscovered after 120 years by Dr. Gopal Krishna Bhatt and his team in Shimoga. And in fact, I called him and I met him for collecting this plant, and he was so generous. He came along with us, and he could show the plant to us. And what we did is we used the ecological niche modeling to understand the distribution and prediction, and for getting the distribution and prediction map of using ArcGIS. So you can see here the distribution map on the left side and the prediction map on the right side. And you can see here with the use of this ENM modeling, we could find 3 new populations. This first time it was not, we published this information also. This is the advantage of using this computer algorithm. And with that, you can find any new population of the plant is available in the nature or not. And we use this technique for other species also, but you should have at least 10 lat long information, then you can put it in this algorithm and you can get the information. And you can see what we did is we developed a prediction map for this species for 2020, 2040, 2060 and 2080. You can see there is some change in the distribution pattern of the species. And next question, are the collected specimen are authenticated? We did one thing, all the collections we made was shown to Dr. Gopal Krishnan, but he is authority on this plant species. He is the person who rediscovered after 120 years. And when he say that it is this plant, then only we taken is that sample as belonging to the Maduka insignis. But again, sometimes he was also confused whether it is same or not. So to overcome this problem, what we did is we developed a marker, and you can see, this species is very similar to another species. There are 5 species of Maduka available in in India, and Nerifolia are also very similar to the Maduka insignis. So it is difficult to identify this species. And you see here the seeds are also very similar in nature. And we developed a scar marker for identifying these species. So it is a species specific marker, and this is how we develop the marker. I'm not going to the details. Next question is how these species can be conserved. As I was mentioning you, it is the population of this species is very rare, and we develop a species specific conservation strategy. Initially what we did is we wanted to propagate this plant in large numbers, and we developed a microbial consortium. This microbial consortium we developed from the soil available the root side of this plant. And we use this, in fact, seed germination is not a problem, but though seed is germinating, the plant is not establishing. And we applied this microbial consortium into the seedlings. It developed very well and it established. You can see the plants which are applied with this consortium, the development was much more. And we developed large number of seedlings using this method, and we approached the Karnataka Forest Department for introducing this plant in natural locality, and they were very considerate and given permission and even all the logistics, and we could re-establish this plant into to the tune of 600 number in different localities of Karnataka. And can see the re-established plant, and now we are monitoring it. And the CPCRA Kidu Center also helped us to re-establish the plant in Kidu, thanks to Dr. Anita. And you can see the it is available in Nisargadama. Also we re-established there. They're having a good population of this species. And in IHR also we have some 25 plants established, and though it is a riparian species, we could establish this plant at our place also. And you see here this only 21 plants available in nature, and the one which is with a red ring we could locate for the first time. And these are the problem faced by this species: excessive sand mining, then fragmentation, And again, in all places, this sand mining is a big problem. Then loss of inflorescence and the damage due to the frugivores. And these are the output of this project, that is a population study. First time isolation characterization and submission of the plant growth promoting fungal and the development of scar markers. Then these gene sequence we deposited in NCBI. Then we did the chemical profiling. We did the reintroduction. Then we updated the IUCN status and providing roadmap for future conservation and defining exciting research areas. And we had a lot of publications came out of this project. It is available online. Now, what is the global status of this genetic resource of horticultural crops? You can see the, in FAO, they have mentioned about the root and tubers, vegetables, nuts and fruits, then medicinal, aromatic and spice crops, and industrial and ornamental plants. And coming to our national gene bank, see, the NBPG are also not having expertise to conserve the genetic resource of horticultural crops. In fact, IHR is much better placed than NBPG. But for the seed propagated horticultural crops, they are having this many gem blossoms stored at the seed gene bank. Then for cryobank, also they are in vitro gene bank and cryobank also having some accessions of horticultural crops. And these are the summary of the ex situ conservation in different geographical region. So now coming to the use part, there are problems with the horticultural crops which I have mentioned, and in 5 minutes I just tell about this, and will close my talk. That the main driving force behind the efforts on conservation is utilization, as all of you agree with me. And it should be done by the crop improvement by the researchers, and adaptation and sustainability needs of farming community. Then collecting and conservation of horticultural resources has made significant progress, and only now we should concentrate on wild relatives, and effective use of them is still waiting. We are not, though we have a lot of collections, the use is very much limited. And some of the constraint challenges are no real consideration of benefits to farmers, and decision on conservation based benefit to the farmers, and fencing of the crop wild relatives may be good for immediate use. And future value may diminish as evolutionary potential, and move part of them to ex situ conservation for utilization. Then most gene banks are yet to characterize and evaluate. Though I was showing so many accessions, these accessions are not characterized. And now all the breeders are looking for trait specific gem blossom. Say such a gem blossom is not available with the NBPG or with IHR. Then incomplete characterization, then limited access, trait specific evaluation still yet to be done, highly limited characterization, and little attention to collecting and researching horticultural species. And as I mentioned, most of them are orphan crops, and lack of efforts on pre-breeding activities. Then gene bank to be able to sell accession by enhancing value, and we should take up the pre-breeding activities. And little research on cost-effective long-term conservation, which I was mentioning earlier also. Little use of modern scientific methods like cryopreservation. Then gene bank curators, managers rarely involved in research. Then increase research on issues ranging from population genetics to policy studies, and watch for new problems of conservation. Use of geographical and spatial information, which I was showing you in the case of Madhuka in Sigris. Increase analysis and use of georeference data from the gene bank is also very important. And integrate conservation of underutilized species. Most of these horticultural species are underutilized. Enhance the studies, but I was telling you it has got a lot of health benefits. Now our food basket is completely changed, and we are taking a lot of fruits and vegetables. So there is a lot of scope for horticultural gem blossom. The lack of refined strategies, and as medicinal aromatic plants are mostly collected from the wild, requires approach of different conservation strategies. Need strategies to conserve and at the same time minimize the exploitation of naturally occurring resources, and restoration efforts may be needed. Now I will just summarize the issues. See, the one issue which we are facing, how to get gem blossom from other countries. After we signed the CBD in 1994, conservation or the conventional biodiversity. After that, the genetic resource become the sovereign property of the country of origin. So with that CBD, we are not getting any gem blossom, especially for horticultural crops from different countries. FAO is administered ITPGR, okay, that is in the international undertaking on plant genetic resources, but they are having only very few horticultural species included in that list. So it is very difficult. We were trying to get colored tuberose from Mexico. We were not able to get because they are asking 50% royalty for the gem blossom. Then cultivation of exotic crops. Now farmers are found cultivation of exotic crops very remunerative. For example, dragon fruit and kiwi fruit. Recently, our scientist Dr. Karunakaran was telling, if you cultivate dragon fruit in 1 acre, you get a profit of 6 lakh rupees per annum. Then kiwi fruit also is having lot of health benefits. People started cultivating. Then use of gem blossom stored in gene banks. We are yet to characterize, and now NBPGR has taken up large scale characterization of this gem blossom. In fact, for 16 crops, the present director, Dr. Kuldeep Singh, got 16 projects from DBT sponsored projects. In that, they are doing the large scale characterization, including the molecular characterization. And such projects will help us to, have these trade specific gem blossom, and then it can be readily used for improvement of the crops. Then use of crop biorelatives. See, if you want to deal with the climate change and other natural calamities and abiotic and biotic stress, the crop biorelatives are the only hope. Then another issue is most of our horticultural resources are in national active gem blossom sites. There are in fact 30 such sites all over India, and there is no funding mechanism for this NACs. And in fact, we were in constant touch with NBPGR, and they have done some preliminary discussion with IHR, and the director of NBPGR is trying to get some funding for this national active gem blossom sites. And ABS, that is access and benefit sharing. Now, if you use any gem blossom from anybody, we should be able to share the benefit with the owner of the gem blossom. But that is not done in India. We are yet to take a call on this. In fact, I was as a expert member of access and benefit sharing in Karnataka Biodiversity Board. I found, though they have sent letters for 300 companies, only 50 companies responded, and 30 companies signed the agreement with the Karnataka Biodiversity Board. So in the coming years, that will become a issue. Now, those who want to know more about the conservation and utilization of horticulture genetic resources, this volume is available online from Springer. In fact, Dr. Anita contributed a chapter in this book. And for conservation and utilization of threatened medicinal plants, another volume is available, and another one more exclusive volume on fruits, where Dr. Mohan has contributed a chapter on cashew, and one on protocols for the pollen curve preservation is also in the making, and next year both will come online. So this is my if you want to know more about the publications, you can go to Google Scholar or ResearchGate, you can find. And at the end, see, 75% of the world's poor are rural, and most are involved in farming. And in the 21st century, agriculture remains fundamental and for poverty reduction, economic growth, and environmental sustainability. And our national food security depends on our ability to conserve all our biological wells. All of you agree with me with this. I would like to end my talk here. If you have any queries or any comments, you are welcome. Again, thank you very much for your invitation. I enjoyed talking to you, and thank you very much, Dr. Anita, and all my colleagues in DCR Puttur and CPCRI. Thank you very much.
Thank you, Rajshekaran, for an excellent presentation.
Thank you.
I know it is a very vast topic.
Yeah.
And concentrating to certain aspect is very difficult. Conservation aspect of horticulture. Horticulture itself is a huge topic, and this conservation. So it was an excellent talk. Hope everybody has enjoyed it. Any queries from our scientist side, they can ask.
Yeah, let me start with one of the queries. Sir, it was a very wonderful talk, and I think everybody talks of conservation and utilization in every other fora. But I think plant genetic resource scientists, we need to be vocal in getting the funding for further work, conservation and utilization. We are not getting as much as fund we want for our work. That is the one observation. And the second, the question I wanted to ask was, what is the importance of Maduka insignis, sir? What is its use for? Is it a medicinal plant or rare or whatever?
Yeah, good question. In fact, I forgot to mention that it is, in South Canara, it can be found on the this river is there. No, that river is flowing from Subramania to towards the other side.
Kumardhara. Kumardhara.
Kumardhara. Yeah. It is found only on the banks of Kumardhara, and the seed is that oil is extracted, and that is having some medicinal value. And we found, we did the cancer studies on the leaf extract, and we got positive results. So it is a medicinal plant, and the local people are using for different purposes. We collected some traditional knowledge also from the people living near these populations.
Okay, sir. Thank you. Any other queries from other participants? Sir, there are about 46 participants. There were 49 to start with.
Yeah.
So 46 participants are there, still there. So they can ask questions if they have any.
Rajshekaran.
Yeah.
You have been storing this pollen since 1984 or 85?
83.
Because I know I was there during that time, along with Dr. Alexander, sir. So, but whether you have done hybridization or using this pollen for artificial pollination?
Yes.
35 years. What was the success of getting the?
See, in most of the horticultural crops, we are lucky to get the pollen in sufficiently dried form. That is why we were able to get success in the pollen cryopreservation. And we used this pollen for hybridization in tomato, in brinjal, then in capsicum, then in chilies, and we found it is at par with the control. Control means we use the fresh pollen, and there was no change in the viability or change in the fertility. Fertility means it was able to set fruit and seeds. There was no change. And in fact, for the fruit crops also, in papaya, then in grapes, papaya, in grapes, mango, also we did, and it was successful. And now, as you are also one of the contributors, now we are compiling all these protocols for horticulture crops, and the protocol book will be published by NBPG, by Springer. And we are just waiting for some more chapters, and most probably next month it will go to Springer. In fact, from Springer, I am getting every month I am getting email asking what happened to that. So
Okay.
there we are compiling the protocol for all the species.
Okay. With regards to mango,
Yeah.
previously we used to conserve it in a anther form.
Yes.
During
See,
1986. Yeah.
mango, as you rightly mentioned, only one anther is fertile. So
Yeah,
what we
remaining
used to
are-
do is, and per anther pollen is 40 or 60 pollen. So we used to conserve in the form of anther only. But when you take out, you can see the pollen is already burst, and you can, in a powdery form, you can use that for the pollination.
But now also you are following the same method, or now you have any procedure to extract the pollen from the anther and cryopreserve it?
You know, the issue is if you do like that, we lose some of the pollen because it is very, very light and very small.
So
we even for some of the other species also now we are storing in the form of anther only. When
so
the
cashew,
pollen
what
is
is
small,
your opinion? Cashew, what is your opinion?
Cashew also we can follow the same
Anther.
procedure. Yes.
Anther only.
Yes, anther is the best.
Okay, that's what I want to know,
Yeah,
because any new method has come, because when
No,
I
no.
did because I did for a PhD, I did mango anther. So afterwards, now
And
30 years
I feel other than IHR, you only taken the pollen cryopreservation in a very systematic manner, and you could successfully transfer the technology to some of the entrepreneurs.
yeah,
So,
we have commercialized
I
the
think
technology.
that is a very good thing.
No, but no other horticulture institute. We have
No, the Dr. Rajan from Lucknow, along with the NBPGR, they have done for mango and published a paper in the Science of Horticulture. That's all.
No commercialization. We have
no commercialization. Nobody has done it.
50,000 rupees. We fixed the price, and many people, 2 people have taken the technology.
Yes.
For coconut.
Sir, we have with us Dr. P.L. Saroj, who was the earlier director of our institute.
Yes.
Is now the director of CIH Bikaner. Sir, if he has any queries, sir, P.L. Saroj, sir.
In fact, Dr. Saroj is also one of the contributors for the volume. He was very kind enough to contribute a chapter for the volume on fruits.
Dr.
Sir,
Saroj,
you are muted, sir. You are muted. You are muted.
Good afternoon to everybody.
Saroj sir, welcome sir. I was not knowing that you are there because I can see only small box.
Many good things he has spoken, and this is also required. Particularly, the need for the cryopreservation in case of a date palm is essentially required, and we are thinking to develop a pollen bank of the date palm.
Excellent, sir. Excellent. Yes.
And why it is required?
Yes.
That most of the pollen material means male plant, their timing is different than the female when they are coming in flowering. So pollen storage is a very, very essential activity. So far we have not yet done. And in refrigerator and some here and there we are storing and we are anyhow utilizing. But a very systematic research for the pollen storage in case of date palm is required.
Exactly, sir. Yes.
But that kind of effort has not been made so far. And major reason is the paucity of the funds. We several times in our EFC, we put that some things should be created, but that kind of facility has not been created. And what happens on farmers field, a lot of area expansion program has been taken place in case of date palm, because government is giving subsidy on the date palm cultivation. So all female planting material they have imported and area expansion took place. But when it came in the flowering, the male plant was not there. So now farmers started complaining that how to go for the pollination aspect. So this kind of problem, which was required to be solved at the beginning, now we started thinking. So this kind of things are there, but this is very important topic, particularly for horticulturists and those who are dealing with the perennial horticulturists. So
already, US
who asked me to put little, but really I had enjoyed your talk and many good things you have given, many things you have suggested, but it required a very coordinated efforts.
Yes.
Those who are dealing with the perennial crops.
Exactly, exactly.
Problems are lesser because of the honeybee and everything that is maintained. But in perennial crops, it is really difficult.
Yes.
So we have to look into in palms, in cashew, in date palm. It is really a problem. We have to think of. And in date palm also, another problem is that this is neither the pollination is neither taking place by the air nor it is taking place by the insect, because there is no aroma at all in the pollen of the date palm. So this is a very typical crop. So we have to think of on such crop. So I think just I am putting my views, but others, as Dr. Anita was putting about the cashew. So this kind of thinking is required and some coordinated effort should be made.
Thank you, sir. Very valid comments and very valuable comments. Thank you so much.
So shall I tell about date palm little bit?
Yeah. No
So
problem.
in case of date palm, it is we can go for cryopreservation immediately, sir. No problem at all. Either in Bangalore or we can have it in NBPGR, because date palm pollen is also orthodox types, not recalcitrant type. I think you can standardize in the season, this season itself, and you can go for the best pollen parent. You can cryopreserve it, so that and from down itself you can. We have a technology at CPCRA. From the ground itself, you can pollinate at the top. We have a device developed at CPCRA. You need not climb the palm.
Okay.
So with that, because the number of pollen we get it from date palm is very huge, because you get 2-3 kgs of pollen. The inflorescence
No, not 2-3 kg.
it is there. The huge inflorescence.
Gram pollen. Whatever the males are available in India, its productivity is only 250 to 300 grams only. It is not 2-3 kg.
No, I mean, I'm telling the amount of pollen we can extract is very huge compared to our coconut and in case of arecanut. The inflorescence is so huge. Lakhs of male flowers are there.
No, lakhs of male flowers are there, madam.
Yeah.
Here, in case of date palm, only Dactylifera we have to use, not the silvestre date palm, which is already available in our country. So therefore, there is a limitation. If we will use the date palm other pollen, then the quality aspect will be affected.
No, we have to conserve only the superior ones only.
Hey girl, want to get a drink before we go?
That we can do it.
Yes.
Actually, I think one of your CAH scientists of crop improvement, one season they can standardize it and cryopreserve it either in your institute, one cryo tank if it is there, and continuous supply of liquid nitrogen is required. With that, they can start it.
Okay.
Now, I think Rajshekaran can tell one or two cans are sufficient.
Yes.
For the area.
Yes.
We are doing it like that, sir. And we have also commercialized the pollen cryopreservation technologies to the entrepreneurs to have been taken.
With less than 10 lakhs, you can start and you can have it.
If you want, we can also give the training as a pollen pollen cryopreservation, because we are expert in that.
I know you both will extend the training.
You're always welcome. Yes.
Yeah.
Dr. Mohan, yeah.
Yes, sir. Yes, sir. Any other queries from the participants? Many scientists, even ECRIP scientists are also there.
Yeah.
Dr. Vikas from Jagdalpur and many
yes,
people are there.
Am I audible, sir?
Yeah, audible. Go ahead, please.
Yeah. So, respected all the directors and scientists from different centers, a very good evening. So my question to sir is about two major crops of our region. That is, one is Chironji, Bushania lanzan, and another one is Sulfi, that is Horticultural urens. So the germination in this Chironji, Bushania lanzan is very less. It's around 5 to 6% what we are getting in our region. After even after treatment, what we are getting is very less. And what we are having is some of the superior genotypes in very interior forest areas. And I definitely want to go for the conservation of those genotypes. Yes, grafting is possible, but what about the other in situ techniques we can go for, sir, for particularly of Chironji, Bushnani and Azan. And then afterwards, I can come to Caryota.
I didn't get your question because your audio is not very clear.
Yeah.
Oh.
Audio, there is some issues.
Why
Mohan,
don't you type?
you can.
Why don't you type your question here and send it to me via chat?
Yeah.
Better. Meanwhile, some other can ask questions if there are any. Dr. Gajbia from Vengurla is also there, and many people I see, but many of them I don't know also. The CPCRI people are also there, and
Yeah.
any other question? I think he's going to type the questions, Vikas.
Rajshekharan, we are just couriering the pollen to NBPGR. The viability is, it can.
Yeah. In
no problem. 15
in
days
palm, there is no problem.
No problem.
Yes.
It is
And
orthodox side.
for this date palm,
It's a dioecious actually.
yeah, USDA, because USDA people have contributed a chapter in that. So there one student from Brazil, she has done her PhD in this pollen crop preservation in date palm. She has contributed a chapter in this book. And in most of these Arabian countries, they are also storing the pollen and using it for, as Dr. Saroj told, it's a problem. So the crop preservation is a real technique, and now we are moving from conservation to use, as you also mentioned, and we also given this training for 5 seed companies. Now they are using for the hybrid seed production.
Because many of the vegetables and all, they are uniformly growing with the use of drone. With the use of drone, this crop preserved pollen can be sprayed morning itself. Before this commercially they are doing it. Western countries. It is possible. And any questions? The chat, it is there. In case of coconut, we have done it for last 8 years. We have stored in the cryopreservation, and we could set the normal nut set. Only 8 years only we could do it.
Yes.
Afterwards, last 2-3 years we could not do it. Otherwise, it is possible. We can cryopreserve. It is not a problem.
Mohan, you can conclude now.
Yeah, sir, I got the questions from Dr. Vikas. I called him.
Yeah.
And he tells, sir, other than cryopreservation, they are facing germination problems in Buchanania lonzan, that is Chironji, and also Caryota urens.
Germination
So
of what?
Germination of seeds.
Okay.
And other than cryopreservation, what are the techniques or things that can be deployed for getting the good germination? That's what he asks.
I have no experience in that species. I think Anita can throw some light on that.
No, I don't know about medicinal plant. I am not at all.
This is not medicinal plant. It's a fruit tree, I think.
Fruit tree, sir. Buchanania lonzan is a fruit, wild fruit, and Caryota urens is your palm.
That is a palm. That is fishtail palm.
Fishtail palm, yes.
Yeah. So what is the problem with the fishtail palm?
They're not getting sufficient germination.
No, that is
no,
maybe
sir.
some.
Am I audible?
Yeah, you are audible now.
Yeah. The thing is that in Chironji particularly, we are facing the problem of germination. But for getting the true to type plant, we have to go for the grafting also. The success of grafting is also very less in those type of this crop. So what we can go for this conservation of this genotypes of this particular crop in our region, sir, other than cryopreservation, as we are not having cryopreservation and
facility. You may not have the facility.
difficult for so definitely we have to conserve those type of germplasm which we are
Yeah.
having. We are having high yield of this minor fruit crops.
Dr. Mohan,
Yes,
what
sir,
is
yes,
your what is your idea about that? What he is mentioning? You have any idea?
I think seed
No idea
germination.
about-
Dr. Saroj was raising his. I think he.
Yeah. Saroj sir, please.
Yeah. Regarding Buchnania lanzan, it is called a chironji plant, which is a dry arid fruit. It is the of same family of the mango and cashew, and pollination is not at all problem. But here seed germination is a little problem. Vegetative multiplication is not at all problem. It is all standardized. So pollination and vegetative multiplication, all things are known, but seed germination is little poor. That we have to think of how to improve the seed germination.
So again, different
He
methods
can-
of seed germination.
Sir, he can contact you for all these details so that they can deploy there.
No problem. Regarding Buchnania, we can, whatever the information is required,
Yes, sir.
we can tell.
Yeah, some use of growth hormones can improve the germination.
Exactly,
Some
that's what I was
different
also thinking. Yes.
Yeah, growth hormones or some clarification or so many techniques are there. No?
Yeah.
One will work. Yeah, if no more.
Are there any questions from other participants? If there are no questions, I request Dr. Ashwati Chandrakumar, who is our extension scientist, to present vote of thanks. Dr.
Sir,
Ashwati?
yes, sir. I think
Yeah.
I'm audible now.
Yeah.
Yeah, audible.
Sir, as we've come to the end of this today's session on conservation and utilization of horticultural genetic resources in India, I take this opportunity to thank our guest speaker, an eminent scientist, Dr. Rajshekharan PE, Principal Scientist, Division of Flower and Medicine Crops, IIHR Bangalore, for having taken time and delivering a very informative talk on the above topic. On behalf of our institute, I extend a sincere thanks for sharing your experiences and findings, sir. Thank you very much. Next, I would like to express my gratitude to Dr. Anita Karun, our acting director, for her constant support and also for attending today's lecture. Let me also take this opportunity to thank all the participants, including the colleagues from DCR, CPCRI, AICRP centers, NBPGR and CIAH, and colleagues who have also come from other institutes, our farmer friends who have taken time to attend this session, as your participation means a lot. Special mention to our former director, Dr. PL Saroj, presently director CIH Bikaner, for attending this session. Sir has always made it a point to attend any of the events which have been organized by our institute. Thank you, sir. Last but not the least, I extend my heartfelt gratitude to Dr. Dinagar Adiga, the nodal officer for this program, and Dr. Mohan, co-nodal officer, Mr. Muturaj, technical officer, for their support in conducting this program. And we hope to come with another informative session in the next week. Thank you all. Thank you, sir.
Thank you.
Thank you, sir. Thank you, Rajshekharan sir. Thank you, Sharan sir. Thank you, madam.
Thank you. So we can leave.
Yeah, I'll close the meeting for all.
Yeah.